welcome back to the third part of our Aerosol and Health session. Now Barbara will we move a bit more from ambient and measurement techniques to the health, and now we will have something about cell models and liquid interface models from Barbara Rothen from Fribourg University. So please, platform is open. We're looking forward.
Thanks a lot, Ralf, and also Hendryk, to organize this session and for inviting me to tell you something about the lung cell models that we are using. I think you are all convinced, and what you heard in the previous talks is that the exhaust is really a very complex mixture. So which means do we have now to assess the health effects of the individual components, but also maybe some mixtures. We already heard something from Ian about animal results, and I will first start to give you, no, this was the wrong. Now I'm okay. So to give you an overview about what we can do to assess the effects of exhaust. So of course we can do epidemiological studies, but here it's very difficult because these are very long-term studies, and it's more difficult then to also say that this effect is based on this pollutant because we have a lot of unknowns and a lot of mixtures. So we can then do more controlled exposures with healthy volunteers in an exposure chamber. Again, this is a great tool. It also takes a lot of time. It's expensive, but the results are really interesting and great. We can do animal experiments, inhalation, instillation, nose only. Again, with some ethical questions, of course. Then we will talk now a little bit more about lung cell cultures, but something else that was done quite often during the recent times was to test mutagenicity with the Ames test, so where you have chemicals, metals that can be dissolved in a solution, and then you can assess this with bacteria. So I will focus now on the cell cultures here as an example of how you can assess emission exhaust in lung cells. So the next thing we need to look into this, and this has already been shown very nicely by Oli, is to see where the particles deposit. So the inhalable fraction of the particles is maybe below 25 micrometers, then the bigger particles are deposited in the nasal trachea region, and only smaller particles below 2 micrometer hydrodynamic diameter can reach the lower part in the human lung. This is now, again, this predicted particle deposition published by Günther Oberdörster and colleagues. Oli has shown a similar graph. This is just a different view. So as said, so the bigger particles deposit more frequently in the upper regions, and now here the smaller particles really can penetrate deep into the lung. But as Oli also mentioned, also the small ones can deposit in the nasal fraction, and then they can penetrate via the olfactory bulb nerve directly into the brain. So we focus now on the lung parenchyma, so the alveolar region, which has a huge surface, 100 to 140 square meter, as it has been shown for humans. And we try now to mimic this compartment here at this air-liquid interface possibilities using permeable membranes. You can then remove, so you culture the cells here on the surface or below the insert, then you can remove the cell culture medium from the upper part, you leave the cell culture medium in the lower part, and with this you generate this air-liquid interface. So we decided to develop this co-culture model based on epithelial cells which cover the lung surface from the nose down deep into the lung. So you can use very different epithelial cells, you can use ciliated cells, you can use bronchial cells, alveolar cells, you can use cell lines, primary cells. So here we have really a lot of choice. And it always depends on your research question and maybe also the compartment you are looking at, which is needed to decide for the cell type. So because we were interested in immune reactions and also allergic reactions, we decided to include macrophages and dendritic cells, which can be derived from human Buffy code. So you can isolate monocytes and then differentiate them into these two populations. Yeah, macrophages are phagocytotic cells on the surface of the epithelium, and dendritic cells, they locate more in the basal side of the epithelium, make these pseudopods towards the luminal phase to capture antigens, and then to migrate to the lymph nodes and to present the antigens to the T and B cells which are there. So they are really key for the allergic immune reactions. So here you see, I'm always wrong. So here you see a live cell image video where we labeled the macrophages in green, and red shows the epithelial layer. And you see how these macrophages crawl on the surface of the epithelium. We also did some experiments with diesel particles or carbon nanotubes, and we really could show that that the particles were taken up by the macrophages. So now this was the system we have established, and then we started a collaboration with the exhaust gas control in Biel, and we actually started with scooter emissions. Then we had some campaigns with diesel cars, followed by gasoline cars. So this was a collaboration which lasted over almost 20 years. So Professor Czerwinski has been retired in 2019, so the collaboration then stopped, and I'm really, really thankful that I can now continue this work in the ULTRAS consortium because I need the expertise, because I really need the expertise from the exhaust people and the analytics, which I cannot do. So I will show you now results from two former PhD students of the team, Sandro Steiner and Christoph Bissi. Christoph also joined Ralf's lab for some years as a postdoc. And what is really nice now with this system is that we could direct the exhaust directly onto the lung cells. So by connecting here the exhaust with this tile pipe here, we could direct it over the cell cultures, and this is shown a little bit better here in this movie. I don't need the noise actually. So this is a gasoline car, and on the right side you see all these machines that you need to analyze the exhaust, and this is now my expertise. Here is this blue box, which we designed with an optimal environment for the cell cultures. So here are the boxes with the cells exposed to the exhaust, and on the left side you also have a chamber where you expose the cells to clean air. It's really important to compare the cells kept at almost the same condition. So with this, we really did a lot of campaigns, and I just show you one example where we looked at the diesel car, of course, with and without particle filter. I don't need to go into the details. You see almost no change for the volatile compounds, but if you look then to the particles, here this is without the filter. We could also sample the particles on a transmission electron microscopy grid. Here you see these nicely formed aggregates, and we could almost filter all the particles with the particle filter. So we then use this setting to look into effects in the cell culture model. We looked at very different endpoints: inflammation, genotoxicity, lung cell morphology, and we almost did not see any effects for the filtered exhaust anymore in comparison to the non-filtered exhaust, but I show you something else, and this is the oxidative stress response. So after 2 and 6 hour exposure, you see a significant increase of the exhaust exposed cells, so exposed to the complete exhaust with the particles. Of course, we then also did the analysis for the filtered exhaust, and here we were really surprised to see that the response was almost the same. This was almost 10 years ago, so we really had a hard time to explain these results to the engineers. And also, one of our financial supporters was a filter company. And they always said, cell cultures, maybe there is something wrong, you did something wrong, but I think now it's really accepted that also the filtered exhaust can cause adverse effects, maybe even more adverse effects. So what we did then, we repeated the experiments with other cars. And you all know how long this takes to install a new car, analyze everything. So here you see the diesel unfiltered result. So here now we looked at inflammation, interleukin-8, a cytokine which is specifically expressed in these cells, a lot of xenobiotics. And then we had 2 different gasoline cars. And we could see also for the unfiltered gasoline cars, there was a decrease, and it was almost similar to the control. So there was no effect upon exposure to unfiltered gasoline car. Then we started to implement different filters. So here we have an uncoated filter for the gasoline car 1, and we again saw this significant increase in a pro-inflammatory gene. Then for the second gasoline car, we used 2 different filters, an uncoated and a coated gasoline particle filter. And here we saw, which was really nice also for the company, of course, that we could show that there was no increase in oxidative stress and also inflammatory genes. So yeah, we really had a lot of discussions with the engineers, with the people in the team. And then I was really glad, I mean, it's not really nice, but I was glad when I saw this study that was coming out in 2019, where they found in humans, in healthy volunteers, that if you expose these human volunteers to filtered exhaust and in parallel to an allergen, that this can worsen the effect in these volunteers. And how did they assess it? I just show one example. So they measured the expiratory volume. So if you do a lung test, you are forced to exhale, and this is the volume that you measure. And this is a parameter of your lung health. And what they have seen here, this is the dark black. So these are now the volunteers that have been exposed to the filtered exhaust, so no particles, in the presence of an allergen, and they saw a significant reduction in this expiratory volume, which is quite a short-term reaction. So the people can recover after this, so this has no long-term adverse effects for the people. So what we have shown is that in combination with very different campaigns, different cars, that at the cellular level, with the immune cells, we could show that the engine exhaust can induce similar reactions, allergic reactions, as we observe in humans. And also with the cell model, we could show that the exhaust, the complete exhaust, can result in a different toxicity than of a depleted exhaust or of single components only. We also tried to add nitrogen, things like this. So this was really, really nice. So some of the effects, not all, but mainly oxidative stress and also some inflammatory genes were increased even after particle depletion. And I think this is really nice evidence, and also what you said, Ian, that we can use the models to complement epidemiological study and also animal study. I also agree it's maybe difficult because we do not have the blood circulation, the lymphatic fluid, but we can help to add a piece to the puzzle. And what we try now with these lung cell models, it's not exactly the same, but a similar one in ultras, just to give you a short outlook. So we will look into the translocated fraction of the exhaust. So if we can observe if particles can translocate about the lung tissue barrier, and maybe also mediators that are released into the supernatant, and if we then expose this conditioned supernatant to secondary tissue models like liver, intestine, or neuronal stem cells, if we can also use this system to look into effects in organs beyond the lung. And just to show you a first result from my PhD, Gao, who is also here in the audience. So he started to work with empty inserts, so no cells yet. We just added diesel particles in the upper part and then measured with very different methods the particles in the lower compartment. So these are all background, and here only for the biggest pore size we see some translocation. So I think we will now complement this also with cell exposures and then also look into supernatant from other campaigns. With this, I would like to acknowledge the group, the PhD students I have just mentioned, all the collaboration partners, the new collaboration partners, and you for your attention. Thank you.
Okay, now I have also come to the microphone. Yes, so thank you very much, Barbara.